Hi guys, today I'm going to take you through the answers to the two packets we've done recently, okay? Starting off with the packet about ATP, adenosine triphosphate, a super important molecule that's going to be very important throughout the whole year, but especially in this unit. So I'm just going to roll through the answers here. So as far as our labeling is concerned, the adenine section, that's going to be a nucleic acid. And so that's going to be this part right here, these two rings with the ends in them, okay, with the NH2 coming off of it. That's our adenine. The ribose is a sugar. And so here's this 5-carbon sugar. And then here are our phosphate groups right here, okay? So phosphate groups, ribose, adenine. What is meant by the tri in the name adenosine triphosphate? The tri means 3. Why? Because there are 3 phosphates. 1, 2, 3. What is the structure of ADP, adenosine diphosphate? If you know the prefix di means 2, then it should hopefully make sense that instead of having 3 phosphates here, you would just have 2 phosphates. Okay. Okay, next. As far as writing this as an equation, what I would do is I would put ATP plus H2O, and then I would have a little arrow, and then I would put ADP plus PI plus energy. If you said ATP plus H2O, the little arrow, ADP plus inorganic phosphate plus energy, right, that's fine too, okay? But the main idea here is, again, this is hydrolysis, okay? Remember, so we're splitting. So we're taking one thing and we're breaking it into multiple pieces. So we take one ATP, water's involved, it comes in, it busts this off right here, and we're left with an ADP. We're left with that P that got, here the phosphate group, that got broken off right here. And when it breaks, it releases energy. Okay. With question number 4 here, what's happening to the atoms? Just like back in unit 1, how we learned, where an H goes to one and OH goes to the other one, that's what happens here. Okay. So this PO3 breaks off right here, and then the OH goes here, and then over here we have a P with an O, and then the H goes on that one. So OH goes on one, H goes on the other, just like with hydrolysis with our macromolecules, okay? We already learned about hydrolysis, right? Hydrolysis, hydro is water, lysis is split. So water splits the bond in ATP. 6A. This is going to result in a net output of energy because we see this little energy zigzaggy over here is released afterwards. So it's an output. 6B, which one has a higher potential energy? This is going to be ATP because energy is released when ADP is formed, right? When you take ATP and break it down to ADP, energy is released. So that means ATP has a higher potential energy because it has more to release. Question 7A, then is it endo or exothermic? It says exothermic because it releases energy. Okay. Question 7B I thought was really confusing and weird, so just skip it. Don't worry about it. Don't focus on it, okay? All right, here we go. Going to question 8 then. If it's reversible, then what we had before was ATP plus H2O makes ADP plus PI plus energy. So we're going to flip that. ADP plus PI, and all that PI means is it's just a free-floating phosphate that's not attached to any of our macromolecules. That's why it's inorganic, okay, because it's not part of one of the life molecules. So it would be ADP plus PI plus energy, and then you can do a little arrow, and that makes ATP plus water. For 6B, this is called phosphorylation. Why? Because you're literally taking a phosphate and you're adding it onto a thing. In this case, a phosphate is being added onto ADP, so it's being phosphorylated. For C, this would be endergonic, okay? If the reverse was exergonic, this one's going to be endergonic, and you need that energy to form that bond between the phosphate and the ADP. Okay, here we go. The ATP cycle for 9 for 9. Hydrolysis would be, oh, okay. So this over here is going to be hydrolysis because the ATP is getting turned into ADP. So this right arrow would be hydrolysis. This left arrow would be phosphorylation because this is where ADP gets turned back into ATP. So the idea here is it's a circle. A molecule of ATP will have the phosphate be broken off the end of it, and then later on it regenerates, the phosphate gets added back on, so that the same molecule of ATP and ADP, like, it just keeps flip-flopping from one form to the other, depending if the phosphate's attached or not attached, attached or not attached. Question 10a, the energy gets used for all of these things right here: muscle contraction, protein synthesis, cell division. For 10b, you might have come up with things like active transport. Remember how we said that ATP is used there? A more specific example would be endo or exocytosis. Any just general movement of the cells or even of their organelles needing to be moved around. That would all be things that you could use ATP for. For number 11, the answer is given to you right here in this box: respiration or photosynthesis. For number 12, they make this analogy between money. So for number 12A, this would be when ADP is phosphorylated. 12B would be when ATP is hydrolyzed. For 12C, this would be like when ATP is made, but just like stored in the mitochondria and not used right away. 13, drawing a diagram. This would be an example of a diagram. Basically, if it's adenosine monophosphate, you're just going to only have one phosphate added to it, right? We started out with 3. Adenosine diphosphate is 2. Adenosine monophosphate is just going to have one phosphate group. And notice, like, here is to the left, but in your diagrams, in your packet, the phosphates are to the right. It doesn't matter, right, which way. It could be flip-flopped either way. It doesn't matter. But the idea here is everything is the same except for your number of phosphate groups is now 1 because mono means 1. For 14, you would expect it to be exergonic because, again, we're still breaking bonds here. So we would still expect it to release energy. Okay, so here they get into a lot of details with some big old crazy numbers. Basically, this just lets you know how much ATP is used, okay? So look at that every day. And so this is why the ATP cycle is so important, because of the fact that, you know, we can't go through 350 pounds of ATP a day, but because it can regenerate itself, this is way, way, way more efficient than our body having to continually making brand new ATPs. All it has to do is take an ADP and put a phosphate group on it, right? Phosphorylates it. So then it goes into 2 different ways you can do this. It's basically 2 different ways the phosphate group can get added. One way is called substrate level phosphorylation, where basically like a reaction happens, and through this reaction, like breaking something apart, that energy release ends up attaching a phosphate to an ADP. Okay, there's another way, which we're going to learn about later this unit, where you've got this big old kind of cascading waterfall effect with electrons and cell membranes. And it's kind of this big old chain, like dominoes chain effect thing. And you get all these electrons moving around, and that ends up putting a phosphorus on an ADP. And most of the way that ATP is made is through this way, okay, through this second one. A very little amount is made through the substrate level. Okay. So for question 15 then, okay, so it's going to interfere with the flow of electrons. Ooh, so that sounds like the second one here, oxidative phosphorylation. So why would it cause someone to lose weight? Okay, so if it interrupts this form is no longer going to work. You cannot make any ATP through electron transfer, which means the only way you can make ATP is through substrate level. Ooh, and how do you do this? Through the breakdown of sugar molecules. So basically, you're going to generate all your energy from your food being broken down. So your food's going to be used up as energy and not stored as fat, okay? Or if you already have fat that's stored, that fat would get burned to do the substrate level phosphorylation. That's why it would help you lose weight. Why would that be dangerous? Well, once you run through your fat reserves, basically your only energy source would be the food you eat, and you might not have enough energy for life functions if you're not eating enough. So that's why. So that brings us to the end of our ATP packet, and next I will go through the enzymes packet. All right, so here we go now, going through the enzymes packet. Number 1, this is lipase and pepsin. 2A, pepsin is active in the stomach, and 2B, lipase, pancreatic lipase, is active in the small intestine. For number 3, we can tell that pepsin is most active at 1.5, okay, because that's where the graph peaks. And lipase is most active at 8, again, because that's where its line on the graph peaks. For number 4, pepsin has a high rate of reaction at 1.5, while lipase is inactive. I know it's inactive because its little squares and dotted lines are down at the 0 rate of reaction. So it's not active at all. And for number 5, it's flip-flopped. Lipase has a high reaction rate while pepsin is inactive. It's at 0. For number 6, okay, so if an enzyme gets exposed to a pH outside of its range, it's going to be denatured, okay, which means the shape of the active site is going to change, which means it's going to have limited function, or if it changes enough, it's going to have no function. Because remember, shape and function are directly tied together. So enzyme structure would change and enzyme function would change. It would either be, depending on how much the shape changed, it would either be limited function, or if the shape changed a lot, no function. Okay, at number 7, I would say 0 to 4 and 12 to 14, because this is where it has 0 rate. So we know for sure to be denatured there, right? For sure. And for 8, I would say 5 to 14, because again, that's where the rate is 0. So we know it must be denatured there. Number 9. Okay, so we got pancreas pepsin that is used in the intestines, and also, pancreatic lipase, and then we also have like stomach lipase. We would expect to know they're not the same structure, okay? Because the stomach is very acidic and the intestines are very basic, right? As we can see, the pepsin and the lipase have very different pH ranges in which they work in. So we would expect that even though it's a lipase, if it was a lipase that primarily lives and works in the stomach, it would need to work in stomach pH ranges, okay? So it must have a structure probably more like pepsin, right? Or at least one that would allow it to work in an acidic environment. For number 10, add the line graph, a line to show it. So we know it's going to be something similar to pepsin. So it should be something that would kind of mimic this. Maybe it's the same, maybe it's a little bit lower, maybe it's a little bit higher. I don't know, but something that would in general mimic this pepsin where it's peaking here in the acidic range. The peak would be somewhere, in this like 1 to 3-ish range. It's not an exact. Number 11. Okay, so if you take an acid, that raises the pH to 6 to 7. Well, let's look here. At a pH of 6 or 7, your pepsin isn't working, right? So what's not going to be digested? Proteins will not be digested. So if you take an acid, that'll help with your heartburn, but it means that for that moment in time, you're not going to digest those proteins. They're just going to pass through your body undigested. All right, model 2. With amylase here. A, we have rate of reaction and temperature. B, we have rate of reaction and enzyme concentration. And C, we have rate of reaction and substrate concentration. For number 13A, optimal temperature, that's going to be where it peaks. And so it peaks right here, 40s right here. It looks like it peaks a little bit shy of 40. 37 to 38, upper 30s, right in here. B. Whew, biological significance about this temperature. Well, this is a hard one. I think if this were in degrees Fahrenheit, it might make more sense to you. It might stand out to you. This peak on a degrees Fahrenheit graph would be right around 98 degrees Fahrenheit. What do we know that's 98 degrees Fahrenheit? The human body, right? And so as it turns out, 98 degrees Fahrenheit is like 37 to 38 degrees Celsius. So it peaks right at body temp. Makes sense, right? Because it's living in our body. Number 14, what causes a decrease in enzyme activity? Well, it's going to get denatured. It's going to get too hot. Its shape is going to change. It's not going to work as well. So number 15 now. So if you're running a fever of over 40, digestion is going to slow down. Okay. So if here's 40, you're going to be somewhere over in here, right? So digestion is going to slow down. Enzymes are probably still working, but limited. And if it gets hot enough, they'll be denatured. And this is why fevers are so bad. This is why if you run a low grade fever, it's okay. But if it starts getting too hot, this is when people get hospitalized, because literally it's going to start denaturing the proteins in your body, and certain body functions are going to stop working. And if enough of them stop working, you can be in really big trouble as far as your body's concerned, okay? So this is why high fevers can be so dangerous. 16, here we go. So if you increase the substrate, the rate of reaction goes up till a certain point, and then it levels off. This doesn't mean it's zero. If it was zero, it'd be down here. This means it's continually increasing, and now it's holding steady. It's reached its maximum rate, its maximum speed, and now it's just going to maintain that speed, okay? Okay, question 16, enzyme concentration and reaction rate. If you increase the enzyme concentration, you increase the reaction rate. Look at that. The more enzyme you add, the faster it can go, which makes sense, right? The more enzyme you add, the more reactions that can actually happen. So boom, there we go. Fantastic. And that would be our reasoning is there's more enzymes available to make reactions happen. So our number of reactions are constantly increasing. Notice here it says substrate concentration is always in excess, which means it's never going to run out of things to break down. So the more you add, the faster it's going to get. For 17 now, we are looking at graph C. So what's this relationship? Well, the more substrate you add, the faster it goes up to a certain point, and then it levels off. One thing to note is this is not zero. Okay, if it was zero, it'd be down here, right? This is zero. This isn't zero. What it means is, here we go. Oh, faster, okay. I'm going really, really fast. I'm going really fast. Okay, now I'm on the cruise control. Whatever rate I was going, whatever speed I was going with these reactions, it's now like leveled off, okay? And why might that be? Well, you're not increasing your enzymes. You have the same amount of enzymes, but you just keep adding more stuff for them to break down. Well, at a certain point, if all your enzymes are busy and they've all got like, think about it like they're all busy and they've all got like a line of substrate there, like they can't work any faster. They're breaking down stuff as fast as they can, but if they're all busy, they're all busy. And so you can keep adding more stuff for them to break down, but they're all busy. So it doesn't matter anymore. They're working as fast as they can and they've maxed out. For number 18, you were just developing an analogy. You're just going to do whatever made sense to you all. Okay. For number 19, graph B, whatever reaches maximum level. No, it should continue to increase as long as there's always excess substrate for the enzyme to break down. It should always go up. All right, getting to the extension questions, which are always harder. For this one, you're drawing a similar graph to this temperature graph. So you would have rate of reaction, you'd have temperature, okay? But you're going to have the peak be somewhere greater than 70, because it says they live in a hot springs where the temperature is greater than 70. So you're going to have the peak just somewhere be 70 or above, right? Okay. For 21A, at this temperature of 95 degrees Celsius, remember the human body is 37. So at 95 degrees Celsius, the human polymerase, and you don't really need to know what this is yet. We're going to learn about it later. But basically, this human enzyme isn't going to work. It's going to be denatured. So the human stuff, not going to work. So if they find this other stuff, this TAC, that polymerase must work a lot better at higher temps than humans. That's all for 21A. 21B, if you used one, it would be denatured, and so it wouldn't work. And so this whole PCR process, which again you'll learn about further later down the road, it wouldn't work. For number 22, a graph. You should have just a little set of axes, and you should have some type of line that just goes up. Okay, so this is for 22. Because you need the coenzyme and you need the enzyme. And so if it's there, they're just going to go up. As one goes up, the other one goes up. So more coenzyme you add, the faster reactions are going to get, because the coenzyme is what allows the reactions to happen. For 23, if there's an inhibitor, that means the inhibitor is going to mean that some of those enzymes aren't always working because they're being inhibited. So for 23, you'd also have a line, but that line is going to be lower. It's going to have a smaller slope than your line for 22. The numbers don't matter. You don't even need numbers on your y-axis because you aren't even given anything. 22 should be a straight line that has a higher slope. 23 would be a straight line that has a lower slope. If you have more coenzyme, you're going to have faster reactions, but if you have inhibitors, that's going to slow things down. Okay, so that is the run through both of those. If you have lingering questions on them, things that still didn't make sense after I went through them, bring those questions to like one of our in-class question ask times, and I will go through them then.